Hi, this is Courtney, and this is my eukaryotic cell division project. An important role of cell division includes reproduction, which occurs in relation to single-cell organisms, often prokaryotic cells, but can occur in eukaryotes too. For instance, an amoeba is a single-celled eukaryote, uses reproduction to pass its genetic information on to a new organism, which is a copy of the original cells. So there's two identical daughter cells that share the same genetic information, which is done in binary fission. Another example of a key concept is growth and development, which could be the formation of an embryo from a sperm cell and an egg cell, which is multicellular organisms donating their DNA, which creates another multicellular organism. Tissue renewal is another part. It's normal for cells to die in relation to accidents or just degenerating, and bone marrow is a good example, and it represents the repair and renewal of cell division. Now for the phases of the eukaryotic cell cycle. The eukaryotic cell cycle describes 4 phases in which a eukaryotic cell participates in self-replication. The M phase of the cell cycle, which is the shortest of the 4, stands for meiotic phase, which includes mitosis and cytokinesis and will be observed in greater detail later on. The 3 other phases of the cell cycle include G1, S, and G2 and fall under the title of interphase, which is much longer than the meiotic phase. The meiotic phase and interphase can be thought of more as broad titles and have sections underneath them. I like to think of the cell cycle as a big umbrella term, which branches off into meiotic and interphase, which then branch off again into the smaller subunits. The G1 phase is known for growth. It's the longest phase in which ribosomes are produced and hence proteins. The S phase is synthesis and DNA replication occurs. The last phase of the cell cycle is known as G2, and then another phase which growth occurs. This phase is preparing microtubules in preparation for the M phase. The M phase, also known as the meiotic phase, is the smaller section of the cell cycle where cell division takes place. During the meiotic phase, mitosis occurs consisting of 5 phases: prophase, prometaphase, metaphase, anaphase, and telophase. Cytokinesis also occurs during the M phase and overlaps with the N phase of mitosis. During prophase, the nucleus is disappearing as chromosomes condense. Prometaphase occurs when chromosomes are visible and have sister chromatids. Metaphase occurs when spindle is complete and the microtubules meet and the metaphase plate. Anaphase occurs when chromatids separate and daughter cell chromosomes move towards the polar ends of the cell, which shorten the centrochore microtubules. Telophase occurs when the daughter nuclei form as well as on the onset of cytokinesis. Chromosomes apart during metaphase. What is not widely known is how the meiotic spindles pull the chromosomes apart. The meiotic spindle develops in the cytoplasm during prophase. Along with this, the meiotic spindle consists of microtubules which are developed during the G2 phase of interphase. Although the meiotic phase occurs in both plants and animals, it occurs differently in both. The biggest difference of mitosis in plants and animals comes from the absence of centrioles in plants. This being said, even in the absence of centrioles in animals, cell division can still occur as a spindle will still form. Regarding animal cells, the formation or assembly of a spindle microtubulus occurs at the centrosome, which is also known to be the area in which a cell organizes its microtubules. Further, in relation to animal cells during interphase, a centromere will duplicate, hence producing 2 centrosomes, which are placed at polar ends of the cell during prophase and prometaphase. Initially, these 2 cells are located near the nucleus. During prophase and prometaphase, spindle microtubules emerge from the centrosome. Following, an astra will emerge from the centrosome after the completion of prometaphase. Chromosomes, as mentioned earlier, have 2 sister chromatids and contain a kinetochore that contains DNA and are located at the centromere. Microtubules will attach to the kinetochores during prometaphase. These kinetochore tubular pairs move to different ends of the cell away from the location of the microtubules till they reach the middle of the cell during metaphase. Remaining microtubules that are now attached to the attached to other remaining microtubules at opposite ends. Anaphase is now able to commence, followed by telophase, which is overlapped by the process of cytokinesis. Cytokinesis also occurs, resulting in two daughter cells, whether that be in animal or plant cells, which both occur in the cytoplasm. Animal cells observe cytokinesis in the form of a cleavage furrow. The furrow presents near the location where the metaphase plate was, which is a contractile ring of actin microfilaments and myosin. The ring contracts until the cell is separated as it interacts with the myosin. In plant cells, there is an absence of a cleavage furrow due to cell walls. Thus, the cell separates into daughter cells when vesicles move forward along the microtubules to the middle of the cell, forming a plate near telophase. During telophase, the cell plate enlarges so it reaches the perimeter of the cell, which splits the parent cell, resulting in two daughter cells. Cell regulation serves the purpose of maintaining normal cell growth and consists of checkpoints such as G1, M, G2, and S. It should be noted that these checkpoints are regulated by proteins. The G1 checkpoint occurs between S and G1 phase. If the cycle does not go through the G1 checkpoint, it reverts to the G0 phase, which no cell growth occurs. The S phase requires a signal from the cell cycle to ensure proper DNA replication and to continue. The M checkpoint is the checkpoint before mitosis. M phase also has a checkpoint that ensures chromosomes are attached to spindle fibers. A cell knows whether to divide regarding signals such as cyclin and CDK. Checkpoints in the cell cycle are often activated by kinase-cyclin bonds through phosphorylation. Kinase, as previously mentioned, occurs in an inactive state, meaning that in order for them to work, it needs an outside stimulus to give it directions in the form of cyclin. For CDKs, or cyclin-dependent kinases, are dependent on cyclin, and therefore their level activity varies regarding cyclin concentrations. G2 and S phase depends on cyclin levels and are much higher. M phase has lower cyclin levels. When cyclin binds to CDK, it forms an MPF, or maturation promoting factor, which allows the cycle to pass through G2 checkpoint. It allows the cell to go into mitosis, or also the meiotic phase. Most cells obey the rules of the cell cycle, some do not. Cancer cells do not obey the rules of the cell cycle. They ignore growth signals and do not stop dividing, even if growth signals are not there. Sometimes gene regulation is defective, and it can be seen in relation to checkpoints in the cell cycle. For example, an inadequate amount of cyclin, meaning no cyclin-CDK bonds, which result in no MPF, which is needed to move forward from G2 phase into M phase, which results in cell division errors during mitosis. Overall, all steps are needed for the cell cycle in order for cell replication, cell division to occur correctly. Thank you for watching my winter project, and bye. Thank you.